Tools of Human Genetics, Part 1, Module 1, Lecture 3. A mutation is defined as changes in the DNA which affect the function that the protein codes for. Diagnostic methods to search for mutations include PCR or polymerase chain reaction, Sanger sequencing, southern blotting, northern blotting, western blotting, and whole genome or exome sequencing. In parts 1 and 2 of this lecture series, we are going to be discussing these methods. Polymerase chain reaction and DNA sequencing, or PCR. The polymerase chain reaction, or PCR, is a method to amplify small samples of DNA into billions of fragments within a few hours. Primers are used to amplify a specific region. PCR creates enough DNA for analysis. After 30 rounds of amplification, a billion copies of a particular sequence is created. Basically, we have a fragment that we cut out that we're interested in. We then separate the two sequences with heat. We use base pairs to now we have two copies from one. We heat again. We do this process. And after a number of sequences, we now can have a billion sequences, or excuse me, a number of these processes, we have a billion sequences. One complication of PCR is that it does not detect deletion. So if there's a deletion in one copy of the gene, that's not going to be amplified. So only things that are amplified are either the normal sequence or a known mutation we can amplify. But we can't actually sequence a gene, and if there's a deletion there, that won't occur either. However, it is a very inexpensive method to amplify gene sequences. The gold standard for looking for specific changes in genes is known as Sanger sequence. This method uses read-through of the nucleotide sequence of a cloned DNA fragment. There are 4 base pairs, A adenine, T thymine, G guanine, and C cytosine, in addition to deoxy analogs of all these 4. Each base pair of the deoxy analog has a different fluorescent tag. When the deoxy bases are used in the polymerase, they can no longer elongate the DNA sequence. So these bases are randomly placed, and in some cases, for instance, normal base pair of T will be placed until the third one comes about, whereas in other cases it happens the second time, and in other cases it happens first. As a result, there are different size fragments created because, as stated, once the deoxy base is used, the fragment no longer continues to elongate, and we can separate based on the fluorescence where these base pairs are seen, and then we can determine what the sequence of the gene is. So we start with A, T, G, C, T, T, so on and so forth. Now what we're going to do here is show you a small video demonstrating Sanger sequencing.
The first method of sequencing the genetic code was devised by Fred Sanger. To sequence the DNA, it must first be separated into 2 strands. The strand to be sequenced is copied using chemically altered bases. These altered bases cause the copying process to stop each time one particular letter is incorporated into the growing DNA chain. This process is carried out for all 4 bases, and then the fragments are put together like a jigsaw to reveal the sequence of the original piece of DNA.
Mutations detected by Sanger sequencing. The type of alterations detected, the first is a known pathogenic mutation. That means a change in the DNA that has been described before. The second is predicted pathogenic. When we look at this and use computer models, we would expect that the protein to be created by this change in amino acid base would result in a non-functional protein. And then my all-time favorite is a variant of unknown clinical significance. There's a change, and it may be a problem or it may not. This is probably the most difficult thing. And then finally, polymorphisms. And the more that we're doing this DNA sequencing, the more we're realizing polymorphisms, again, we're all 99.9% similar, but there's small changes between us, and most of them do not cause a problem. DNA sequencing, the sensitivity is 99 to 100% for all nucleotides changed in small deletions and insertions. This cannot detect larger deletions and duplications greater than the size of PCR, and it also needs to be targeted to the region of interest. So we need to know what we're looking for. If sequencing doesn't detect a mutation, either the patient may not have a mutation in the tested gene, the patient has an alteration that cannot be detected, for instance, a large deletion that was missed by this, or the patient has an alteration in the gene that is not sequenced, for instance, in intron or promoter regions of the gene that we're not actually looking at. Study question. Polymerase chain reaction PCR can do the following. Amplify a small amount of DNA into large amounts for analysis, identify a large deletion, identify a small deletion, determine unique sequence changes, or analyze changes in non-coding regions. The correct answer is A.